Hello, this is the third video of the series, and today we're going to be talking about metabolism and enzymes. So let's talk about energy first. Energy is basically what life is built on. It's built on chemical reactions, and this is basically driven from energy. And from an ecosystem, it's basically energy is transformed from one form to another. Metabolism. So these are just the chemical reactions of life. And basically, metabolism will form bonds between molecules, which includes dehydration synthesis, like we talked about. We remove a hydrogen to form a polymer. Synthesis and anabolic reactions. Breaking bonds between molecules, which include hydrolysis, like we talked about, which is where you're adding a water molecule to break down a polymer. Digestion and catabolic reactions. So anabolic reactions actually form bonds, and catabolic reactions break bonds. So this is what we talked about before about dehydration synthesis and hydrolysis from our previous episode. Basically how hydrolysis will break down through addition of H2O, and dehydration will form through removal of H2O. So now we're going to be basing and classifying chemical reactions. So some chemical reactions will either release energy, and then some will require an input. So reactions that release energy are known as uh, exergonic, exergonic, and they basically digest polymers, and mainly they're going to be basically hydrolysis reactions, where basically you're breaking down a polymer, or catabolic reactions. And this is mainly because digesting molecules equals less organization, which basically means you're at a lower energy state, so you're going to release energy. Uh, and now some are going to be requiring input of energy, and these are more known as endergonic. And basically, the input of energy requires building polymers. That's basically through building polymers through reactions such as dehydration synthesis, you'll require input of energy, and they're also called anabolic reactions. So over here we have our two types of reactions, and as we can see the difference between here, delta G is the difference of free energy, and that's just basically the difference of where the energy starts, where it ends. So as we can see for exergonic, there's actually a negative delta G. Well, for endergonic, there's actually a positive delta G. And basically over here to our left, energy is going to be released. As we can see here from the negative delta G, we have more energy. And then we actually start off with a lower energy state because energy is released. And over here, since we're adding energy, we're actually going to be breaking this thing to different products. And we're going to have a positive delta G here. So actually, you're going to be adding energy. And that's what forms this free energy diagram over here. So we're just going to talk about some laws of thermodynamics, just the basics of, so we can understand enzymes better. So the first law is that energy can be transferred and it's transformed and transferred, but it can't my bad. Energy can be transferred and transformed, but it cannot be created or destroyed. So what this basically means is that for example, when I move my hand, I'm not I'm transferring energy. I'm not destroying the energy by like moving it or wasting the energy. It just mainly like turning it the energy is maybe transferring into heat, thermal energy. So like for example, when you like do actions, you're not actually wasting your energy. You're just transferring it to the environment. The second law is that every energy transfer increases entropy. So entropy is just a molecular disorder. So basically, every energy transfer that happens in a system will increase the disorder of the universe. And entropy is just the disorderness, basically, or the measure of molecular disorder or randomness. So you don't need to know this equation. This is mainly helpful for chemistry, but just delta G relates to the basically the total entropy, which is or change in entropy, which is delta H, and delta S, which is the uh oh my bad, which is okay. So delta H, which is the change in total temperature, and I mean not temperature, energy, and delta S, which is a change in entropy. And entropy is just the molecular disorderness. and T, which is the temperature. So delta G will be negative for all spontaneous reactions. Spontaneous reactions doesn't necessarily mean quick, but it mainly means that it's gonna uh be favored in the reaction to form a lot of products. So basically, if it's negative, you know it's spontaneous, and if it's positive, you know it's delta G is positive, then you know it's not spontaneous. And you don't need to know this really, but just a good thing to know if you don't know the equation, because delta H and delta S and delta T will all relate to delta G. So this just kind of relates, I guess, all what we talked about. So energy and life, organisms require energy to live, and where does that energy come? That's mainly from coupling exergonic reactions with energonic reactions, which need energy. So now we're gonna actually talk about catabolism and anabolism. We'll go more into specific processes just photosynthesis, cellular respiration in the next few slides. But over here is just a simple diagram. Catabolism is energy yielding because we're actually breaking it down, the energy sources. And over here we're building, so it's going to be biosynthetic metabolism because we're going to be building what we want. Like photosynthesis is an anabolic reaction because we are building glucose. So we this just breaks down anabolism and catabolism. Basically, the delta G values. Since we know that anabolism will increase enthalpy or order, we know can tell that delta G is positive, and since delta G is positive, our Gibbs free energy diagram will look like this, like we talked about, to where the products will have a higher energy level than the reactants, or the more free energy than the reactions, not energy level. And over here, the reactants and the products for catabolism, the reactants will be less, have less free energy. The products will have less free energy than the reactants, which is why we have a delta G less than 0. So what drives reactions? If reactions are downhill, why don't they just happen spontaneously? Well, as we can tell, reactions just drive by reactions. So, by reactions such as when energy is released, basically the Gibbs free energy as it changes, it decreases from reactants to products. Since covalent bonds are stable bonds, reactions are just not going to go down. Reactions are not just going to stop. Since our covalent bonds are stable bonds, they're gonna basically we have to break them down and put energy into them to break them since they're actually strong, and that's where energy comes in. Energy will be released and formed based on how we break, because if we, for example, if we have covalent bonds, we don't break them, we will require energy. Sometimes we need to remove energy, however, to form bonds. Activation energy. So activation energy is just the energy needed to overcome the initial barrier to the reaction, and basically large biomolecules that are stable and must absorb energy to break bonds. Just basically just states that for a reaction, we need energy to start the reaction. Okay, so activation energy. So too much activation for life. So activation energy is just the amount of energy needed to destabilize the bonds, like we talked about. And it's to move the reaction over an energy hill. That's kind of like the barrier that I talked about. So if, for example, if that's the hill over here, like on the top of the red thing, the activation energy will need to be larger than that. So change in activation energy or just the activation energy will be the distance from the reactant to the top of the hill. So catalyst. Catalyst now actually will change this diagram because we're going to be reducing the amount of energy to start a reaction, reducing the activation energy, and because of this, we'll increase the reaction rate. So instead, for example, our new activation energy will be this, and will be way shorter, so less, very, very less activation energy. But uncatalyzed reaction will have a way bigger activation energy. So this just compares the uncatalyzed and the catalyzed form. So what's a cell got to do to reduce activation energy? Basically, like we said, enzymes. Enzymes actually perform catalysts. I mean, what am I saying? Enzymes actually catalyze reactions. So for if we have enzymes, it'll actually reduce the activation energy in a reaction. So over here, this is mainly chemistry again. The transition state is going to be here to basically just when it's the stage in reaction on the top of the hill, and or like we said here, the EA is between the top of the hill to the level line where the reacted star, and like we said, the delta G is here. So this is just a question for yourself to answer this to test your understanding. Okay, so enzymes. So now we're going to go more into enzymes. We talked about the chemistry kind of relating to it. Enzymes are biological catalysts in our body, and they can be proteins. RNA is also kind of an enzyme that many people don't know. It's a ribozyme. Facilitates chemical reactions. So, like we said, they increase the reaction rate, make it go faster by basically decreasing the activation energy. And a good thing to know about enzymes, they do not have changed the free energy. So the free energy will be constant between the uncatalyzed and the catalyzed reaction. And it's actually highly specific because so different enzymes actually will target many different cells or substrates. So basically, not an enzyme that's going to work for one substrate might not work for another because it's also depends on the shape. So we're gonna go some vocabulary terms. Substrate. So I just use this term substrate. Substrate's gonna be the term that the reactant binds to. Uh, it's the term for the reactant which binds to the enzyme. That's the substrate over there. Product. Product is the formation of after the enzyme substrate reaction. So that's our products. And active site. Active site is where the substrate is gonna bind to the enzyme. Okay, so properties of enzymes. Main thing to know is that enzymes can get denatured easily by temperature, pH, and salinity. Any conditions that affect the protein structure will just denature the enzyme, causing it not to work. Enzymes are not consumed in the reaction. This means that enzymes can be used for multiple times. So after maybe one reaction, it'll be going going on and on and on. An actual reaction will maybe take 1 second or less than that in your body, and enzymes will just keep catalyzing substrates and forming products and just keep on going. A single enzyme, for example, can catalyze thousands or more reactions per second, and it's reaction specific, like we talked about. Each enzyme will work with only a specific substrate. Naming conversion. So these are just a few common enzymes maybe you should know. Pepsin breaks down polypeptides. This is in your stomach, and then DNA polymerase. This is the main enzyme involving in DNA replication, like lipase. Lipase will break down lipids. It can happen. It's produced by your pancreas and also by salivary glands. Proteases are involved in break down proteins. They can be involved in apoptosis. And sucrase which breaks down sucrose in your digestive tract. This is just a lock and key model demonstrating basically the enzyme substrate complex. So once the substrate binds to the enzyme, it'll bind to the active site. Basically, the enzyme weakens the bonds between the substrate. That's the catalytic activity, actually. And then after that, the substrate is converted to products. Induced fit. This is a bit more different. It's actually more accurate because we have a 3D structure of the enzyme and how it fits in the substrate. And the substrate binding actually causes the enzyme to change shape to better fit the substrate's shape. And this leads to a tighter fit. It's known as conformational change. So how does it work? There's some mechanisms for it to lower activation energy, speed up the reaction. The active site will actually orient the substrates in the correct position. And for synthesis digestion, active sites bind substrates and it'll put stress on bonds that must be broken. So here are a few common factors that affect enzymes. Cyanide. Cyanide is a very bad compound for enzymes. We're gonna talk about more like hormones and stuff that'll affect enzymes. Enzyme concentration, substrate concentration, temperature, pH. Temperature, pH will cause denaturation. Salinity also might cause denaturation. Activators and inhibitors. These all influence the regulation of enzymes, and we might go into depth more about these. If you want to go more depth about these, you can search up enzyme kinetics. They're more complex, but you can learn about the enzyme concentration, substrate concentration, and how it relates in a graph. So, so here's the enzyme concentration. As enzymes increase, the reaction rate will also increase. But as the enzymes become more saturated, we can tell the reaction rate just levels off, and that's when it becomes too concentrated. And substrates could become the limiting factor. So this is the same thing. Reaction rates level off as substrate concentrations increase, and the reason why initially the reaction rates increase is because as we have more substrates, there will be a more bigger chance of collision, meaning enzymes will actually find the substrate and induce reaction. So this is also a diagram of temperature reaction rate. Temperature actually will increase reaction rate to a certain point, but to the point where it's at the top and cross that, it'll start denaturing and fall rapidly. That's at, for example, in this case, 37 degrees. So this is basically what we talked about. Cold heat increase beyond optimum T will actually disrupt the whole thing. But actually, as we increase temperature, the reason is, as we increase temperature, your molecules will move faster. That means there will be a greater number of molecular collisions in the system. So that means more chance of reaction. So different enzymes like different temperatures. For example, pepsin, for example, like we talked about earlier, likes kind of a more acidic environment, while trypsin likes a more alkene environment or a basic environment. The same thing can be talked about for temperature, right? So enzymes might want a higher temperature, while other enzymes might live in a lower temperature. So like this is the exact example that I talked about. Pepsin likes an acidic environment, while trypsin which actually breaks down proteins, might want a more basic environment. So this just talks about more about how pH will affect the enzyme. It just disrupts the bonds by adding or removing hydrogen ions, which disrupts the 3D shape. So salinity can also affect, which is just salt concentration. That's just something to know. So this is how salt will affect it. It just adds and removes cations and anions, which disrupts bonds and mainly disrupts 3D shape. If anything disrupts the 3D shape, that's the main thing of an enzyme. It just won't work anymore. Here are some cofactors and coenzymes which kind of activate or help enzymes and induce speed up or induce the enzyme to actually catalyze their substrate better or more efficiently. So some cofactors common is magnesium, potassium, calcium, zinc, iron, copper. Non-proteins also can be coenzyme. Cofactors are actually just inorganic compounds and ions. Coenzymes are non-protein, but they are organic, while cofactors are inorganic. Many common vitamins is niacin and riboflavin. I think they do NAD and FAD in our body, so they are coenzymes mainly involved in a lot of metabolic processes, which include cellular respiration. Inhibitors. So molecules can reduce enzyme activity. These are the four main types, which I think we'll go over. So competitive inhibitor will bind to the active site, which just blocks out the substrate. So substrate can't go anywhere now because it's a competitive inhibitor attached to the enzyme. So examples are penicillin blocks enzyme bacteria. Penicillin is a common antibiotic used for strep throat. Uh, Disulfiram, it treats chronic alcoholism and basically also a competitive inhibitor. Non-competitive is actually different. It'll bind to the enzyme separate from the active site, and that site is known as an allosteric site. So non-competitive inhibitors are known as an allosteric inhibitor because they don't bind to the active site. But binding to the enzyme will actually cause it to change shape, so the substrate can't bind no more. So, for example, it causes conformational change, and the active site is no longer functional binding site for the substrate. Mainly, anti-cancer drugs are non-competitive inhibitors, and Cyanide poisoning can is an irreversible inhibitor of cytochrome C. This is a thing you don't really need to know this, but how a rate of reaction depends on the type of inhibition. This is kind of more about cell kinetic or enzyme kinetics, like I talked about earlier. So irreversible inhibition, like we talked about, like cyanide is kind of an irreversible inhibition. Basically, they can be allosteric or also competitive. Sarin is also nerve gas. These are all irreversible inhibition. Cholinesterase inhibitors, where they actually you can now not break and the neurotransmitter acetylcholine because cholinesterase will actually break down acetylcholine. So allosteric regulation, basically conformational changes by regulatory molecules. Basically, it's where maybe an activator or inhibitor will bind to the enzyme, and basically it can either activate or inhibit the molecule, and it won't bind to the active site, it'll bind to allosteric site. Cooperativity is actually interesting. The substrate acts as an activator. So this is actually common in hemoglobin, where an oxygen ion, the first oxygen atom the hemoglobin will carry, it'll bind to the hemoglobin, and that oxygen will actually increase the affinity for the enzyme to bind to more oxygen. So yeah, that's an example. First O2 binds, and now it'll be easier for the other O2 to bind on it, thanks to cooperativity. And that is all. Thank you for watching.